Bacteria are single-celled prokaryotes. Prokaryote meaning before the true seed being nuclei. So bacteria are devoid of nuclei. They do not have nuclei. And therefore, they're going to reproduce, when they do reproduce, via binary fission because they do not have nuclei. When they do accrue genetic variation through their lifespan, it could either be due to mutations or DNA exchange. So they could be receiving DNA from other organisms, such as other bacteria. We would call that second part, the partial exchange of DNA from one bacteria to another, horizontal gene transfer. So the first type of horizontal gene transfer that you can get with bacteria is conjugation. So conjugation is going to be the exchange of DNA between two living bacteria. So first off, the donor is going to form this organelle called the sex pilus. That sex pilus is going to be this long hollow tube, and it eventually connects with another bacteria. And at that point, we would call this the conjugation tube, linking those two bacteria together through a hollow tube. And then what happens is the donor organism, the donor bacteria, will send some of its DNA over to the recipient. And then once that DNA is taken into, the cytoplasm, what's going to happen is crossing over is going to occur. So we're going to have enzymes, typically when we experience, crossing over within our cells, when we're undergoing, meiosis, for example, enzymes are going to be, orchestrating the process of crossing over. So you're going to have, you're going to have enzymes that are going to orchestrate that process. So crossing over is going to occur, and you're therefore going to splice some of that donor DNA into the recipient genome, and then when it divides, well, it's dividing the donor's DNA. Another way that bacteria can carry genetic material. So you may have learned this from either this course or previous biology courses, but typically your bacteria are going to have large circular, circular DNA. So this is going to be, the bacterial genome. But sometimes those bacteria could also contain smaller circular chunks of DNA, and we would call those plasmids. And so the plasmids, those can be shot through the conjugation tube, and they are just small circular DNA molecules. This ORI here, this thing labeled ORI, is just the point of origin. We'll talk about that more in detail later. And what these plasmids will do is typically they contain machinery, or they code for machinery that allows them to continue to replicate. And also, within these plasmids, they're going to contain genes that are going to influence the cell to create the sex pilus and the conjugation tube in order to spread itself. So in a sense, it's kind of like a self-motivated piece of machinery within the bacteria that is motivated to replicate itself and spread between bacteria. So that's conjugation. Conjugation is number one. We're going to talk about three different types. The next type is what we would call transformation. So this is when, let's say, for instance, a bacteria dies, and there's fragments of naked DNA hanging out extracellularly. Well, sometimes what will happen is a bacteria will take up that naked DNA, that naked extracellular DNA, and then just incorporate it into its own genome. We would call that transformation. Transformation is picking up naked DNA. Now the last type is called transduction. This one's a little more complicated because this involves another type of piece of machinery, what we would call a bacteriophage. So these bacteriophages are just viruses that will specifically infect bacteria. And bacteriophages will not only inject their own DNA, but when they are produced within the cell, so you'll have a virus, it'll inject its viral DNA into the bacteria, and then it'll hijack that bacterial genome or the bacterial machinery to produce more of that bacteriophage or produce more of that virus. And when you produce more of that virus, it may pick up, or it may contain viral DNA, but it could also pick up bacterial DNA. And those viruses may then inject that bacterial DNA into another bacteria, which will then take up that DNA and spread horizontally genes from one bacteria to another through the vector of a bacteriophage. So we would call this transduction, where bacteriophage is in a sense the vector of that gene transfer. So here's just a little chart to kind of show you, review. So transduction, that's going to involve, that's what we just talked about, that's going to be bacteriophage. So it's going to require a viral vector. Conjugation is going to be the closest thing to, sexual reproduction that bacteria get. That's where they form that conjugation tube and they shoot some of their DNA directly from one bacteria into another. And then transformation, that's where a competent bacterium here, will pick up naked DNA floating around extracellularly and then integrate that into its own genome.